pay attention. Our next speaker is Dr. Janet Sullivan from the University of New Hampshire, and we'll have 15 minutes of talk plus questions.
Thank you, Lisa. I'll just step out in front of this thing so I think I'll do better over here. Thank you all for coming today. I think that there are a lot of us who know that these digitization projects are ongoing, but I realized today in the poster session that there are people who don't know that these digitization projects are ongoing, or they don't understand the scope of the digitization projects. So I think this is a perfect venue because you've seen a couple of posters about the projects, and Barbara Tears is going to be talking a little later in the afternoon about digitization at the New York Botanical Garden. The University of New Hampshire is participating in several of these different projects, and our data, our Herbarium data, are available in several different portals. If you're familiar with the Consortium of Northeastern Herbaria, the New England Herbaria, and adjacent Canada and New York, New Jersey, Pennsylvania are part of that consortium, but there are similar regional consortiums throughout the U.S. Canada is digitizing their specimens and have their own mechanism for making that information available. And then there are different taxon groups or more special interest portals. So the macroalgae herbarium portal that you see here, the welcome page, the home page, the Consortium of North American Bryophyte Herbaria also has their own portal. Lichens have their own portal. So there are a number of different ways that you can access the information that are available in herbaria today. And a lot of these are ongoing projects. So what you find in the portal today might be different than what you find when you're teaching your fall class or you're in the winter organizing yourself for your next field season's research. Just keep in mind that not all of the data are available now, or maybe only partially available because of these projects all being ongoing. So as I said, UNH has herbarium data available in the Bryophyte portal, in the Lichen portal. Most of the specimens in the Hodgson Herbarium at UNH are vascular plants. It's only our New England vascular plants that are available in the CNH portal for now. And then also we are the primary institution on the macroalgae digitization project, and that's digitizing North American herbaria, U.S. herbaria, digitizing their collections from throughout the world. So these are National Science Foundation funded projects for the most part. So it's U.S. herbaria that are being funded in these efforts. And you'll see that these portals that I have up here, the home pages all have a similar look. A lot of the portals use the Symbiota platform, so you get comfortable with one of the portals, and it's easy to make a transition to using a different portal, either for a different region or for a different group of organisms. And you'll see there are some slight differences between the different portals, but in general you get comfortable with one, and the mechanism is easy to make the transition to the next one. And so basically digitization involves capturing three different aspects of the plant specimens that are in herbaria. You do a search, for example, on the garlic mustard, Alliaria petiolata, and you'll get a list there, in the background there, the list of herbaria that have specimen data on the CNH portal right now, and you can see the herbarium is identified by a little icon there on the left, a little symbol, and then just the skeletal data available and a place where you can click and get the full record details. Right now, we are putting up just skeletal records, but you also get an image of the specimen. So if you really needed to see more detailed information about habitat or location, you can bring up the image, you can magnify that and read the label data for yourself. And then the third aspect is there in the lower left is mapping, georeferencing, so that we can do a mapping of the distribution of all the specimens that are in the portal. And again, keep in mind that this is an ongoing process. So our specimens are up in the portal, our images are up in the portal, but ours have not been georeferenced yet, so ours don't have aren't reflected on that map. But if you're on the website and you bring up that map, you can use your cursor and wand over one of those little dots, and a little window will pop up that will tell you which specimen that dot represents. So kind of cool, lots of different ways that you can access information, get information out of these portals. And this has obvious research implications for those of us who are working as botanists, as field botanists, as systematists, interested in floral diversity and biogeography, or making historical comparisons. This is a page, a specimen record page from the macroalgae portal of Codium fragile, that invasive green alga that's turned up in the northwest Atlantic, the northeastern U.S., just in my lifetime. And then there's a map, and again, that was when we were very first starting to georeference. So you can track the spread of invasive species, track the changes in phenology, lots and lots of possibilities for using the data in these portals. Basically, anything that we have done with herbarium specimens, this gets us past that having to either borrow specimens or go to herbaria, spending all the time of going through specimen by specimen. It's easily searchable and then available, and then you can go on to answer the more complex questions that you're interested, that your research involves. At UNH, we have 4 classes that routinely come into the herbarium and get my dog and pony show, as I call it, my herbarium tour. Depending on the class, I go into more or less detail. Usually it's a class that are using specimens for some purpose or making specimens of their own as part of their class activities. And so it's a way of helping them connect to the bigger picture of what would happen to their specimens, where would their specimens go, why are they valuable, and where do all those descriptions and all the information that's in the keys that they're using to identify their plants, where does that all come from? So as I say, different classes get different levels of information. Usually they come in, they have their tour, and then they go off and do whatever else they're going to do in lab. I teach the systematic botany class, and so of course I want my students to do a lot more in the herbarium. And we spend a whole lab session doing activities in the herbarium just once a semester, which I realize isn't a lot, but that's what we have. And one of the things that I have them do is process their own, or process specimens that have been collected, so they do a mounting, but they do what I call the herbarium treasure hunt. So after they get the introduction, the dog and pony show, then they divide into small groups. I have just 2 or 3 or maybe 4 students in a group, and they get a couple of pages, a little scenario, and they look things like this. So you're planning on opening your own garden center sometime after graduation. You'd like to specialize in native species. You know that rhododendrons are popular, but the leaves of evergreen species are easily damaged by the winter conditions in New Hampshire. You investigate the deciduous rosebud azalea. So what I've given them is a setup. I've given them a common name, and then they have to, and I usually give them a picture too, because they're still getting oriented to the 3D, making that transition from the flat, dry plant to the 3D. And I have a number of these because students have all different kinds of interests. So you want to make cranberry sauce for your family now that you've had systematics, and so where do you find cranberries, they have to find witch hazel. I have people who are interested in dentistry and medicine, and people interested in sustainable agriculture and landscaping. So I have a number of different kinds of things, specimens that they have to find. And then they take that common name, the rosebud azalea, they take the book that they're using in class. No apologies, I use Gleason and Cronquist for my class. They have to look up the common name, figure out what the scientific name is. Then we're making the transition to organizing everything in the herbarium according to the Flora North America. So they have to figure out, has the name changed? Has it been published in Flora North America? If so, that's where they're going to find it in the herbarium. What family is it in? Then they go to the herbarium, they find the specimens, they spread them out, then they answer the rest of the questions. So it might be questions about habitat. Where do they go to collect cranberries? What month would they want to go out to collect fiddleheads versus cranberries? Does the deciduous rosebud azalea grow well in New Hampshire? Do we have a lot of representatives, and does it look like it would be a nice thing for an ornamental? So they have to answer those questions based on the specimens and based on the label information, and then they share it with the rest of the class. And I also introduce them to the portal, but up until about this year, we haven't really been able to use the portal because there haven't been a lot of data available in the portal. And so I usually use another, like Southwest Environmental Information Network, or whatever that, SciNet, whatever that's called, stands for, or a different portal to demonstrate, and here's what we'll be able to do in a few years with New England specimens. Well, now we're in the 3rd year, we're coming to the end of the 3rd year on the Consortium of Northeastern Herbaria grant, and so a lot of data are available. Skeletal data, but still a lot of data and the images available for them to do the rest of the information. So, rather than having them go through the specimens, which is an important activity, it's important for them to have that, to know what the specimens are like, to know how to handle them, things like that. But also, if I have them actually do the activity in the portal, then we can go on to much more detailed and complicated problems, and they can also relate to this a little bit better than they can to the flat, dry plants in the herbarium, where they may or may not be allowed to come in and just play and browse and do whatever they want. So I could have them just bring up the Consortium webpage, and the big arrow there is mine. It's not part of the Consortium because I'm not sure how well you can see this, but there's, on that left-hand menu, there's a search collections, and so they would click on that, and then they would get a list of collections that have contributed data to the portal, and you can see the names of the herbaria, but also the little icons, the little images, and then I can have them say, I can say to them, I want you to only search the Hodgson Herbarium collection, so that there's a button up there, a box at the top that you can click or unclick, search only Hodgson Herbarium collections, or no, I really want you to search everybody's collection depending on what I want them to get out of the exercise. And then they click the next button, and we'd go on from there. And I'm doing this because I realized that this working your way through the portal is not intuitive to everybody, because a lot of people come back and ask me questions afterwards. And I've made a lot of mistakes along the way and gotten lost and thought, how did I do this last time? So I hope this is helpful. Another class that's come into the herbarium, we have a new class just within the last couple of years at UNH, is a team-taught aquatic botany, and Chris Neefus was teaching the higher plants part of that class. And so he thought, have them go into a search page, so have them choose, I only want them to search Hodgson or any number of other herbaria, and then you can enter whatever search parameters you want. So for this class, they're all sophomores. This is their introduction to botany, outside of their introductory class, which was introductory biology. And so that he would have them choose New Hampshire in the state field, and then he just chose habitat, so marsh, pond, bog, some wetlands habitat that might be reflected in the locality information. And then a list of plants comes up, and you can see here they're all University of Connecticut plants. Our data aren't in by that level of, that fine level of detail now. We're just in by the level of municipality, so town, so unless it's pond or bog in the name of the town, that wouldn't come up. But then you can choose, all right, I want this group to work on this plant, I want this group to work on this plant, so he's got here GM Valley, and if you click on, this is where a lot of people get stuck, if you click on the name GM Valley, you're going to come up with a species page, and it's usually the description in Encyclopedia of Life or Flora North America, but if that isn't up yet, you're going to get enough information not available, and then you'll think, oh, there's no information available other than what was there. So what you need to click on is the full record details, that second arrow down there, and then you get the full record details, you get the image of the specimen. He has them go to GoBotany, the website GoBotany, so they can see what the 3D real plant in the field looks like, and then again you can bring up that map for distribution. And then he has them ask questions like, where else does this species occur? What are some associated species? Is this a bog or more species? Does this only occur in this habitat? Is it a good indicator of this habitat? For more advanced classes, and Art Matheson isn't quite as comfortable with this portal yet, but I foresee him doing this. In 2003, he published a paper in Rhodora on that Podium fragile. And it was introduced to the Northwest Atlantic, the northeastern US, in 1957. And by early 1980s, there were only about 66 collections. His paper covered the years to 2001. He searched collections, again, going through herbarium specimens and doing the detailed work that way. I searched and then wrote this paper on the expansion of this invasive species. I searched, since his paper was published, up until last year, how many new collections are there? And there were about 74. So you could have your class go and do a follow-up study to this, a more detailed follow-up study. So there are lots of ideas. I'm sure you have lots of ideas of how this could be used in your research, but also if you're teaching, used in the classroom. The students have lots of ideas. If you go to the CNH portal, you can see that Rutgers has a flora of their college natural area. People are asking me about a flora of college woods all the time. We could build that and have it available in the portal. They're interested in invasive species. And if people know the value of the collections and they can use them in their research, whether it's a class project or a senior thesis research project, then they're going to add to the resource. It's a very rich resource, and these portals make a very powerful tool for making that resource available. Thank you.
Great, we have time for some questions.
Are you familiar with New England, Indiana's.org?
Right. And Ray is contacting me all the time saying, this specimen is a county record and you can refine the locality information. I know where this site is. Before a lot of our images were available, we had sort of a clunky way of getting everything up into the portal, and so for a while we had data up without images, and then he would contact me and say, oh, you have this specimen, can you give me the habitat information? So now all that's available, and now I'm getting more data refinement from him, which is great. So if you're familiar with locations, sometimes a lot of these older herbarium specimens especially have abbreviated locations. A lot of our specimens from Wolfboro were collected by someone who worked at the academy there, and they're abbreviated as Wolf. And so they don't even say New Hampshire. And it took me a little while to piece together thank goodness for Google, who this person was and where he was living and working, so that I would know, oh, he's talking about Wolfboro, great, Wolfboro, New Hampshire in Carroll County. So if you see specimens in the portal and you know more information, that's a good way that you could add, and also just refining the information that's available, and it makes it a much more valuable resource. Yeah.
Real quickly, as you're talking about the locations, rare plant locations, is that
They're blocked.
Okay.
They're blocked. You know, some people would like to block just everything, and well, we relied on the Natural Heritage Bureau to tell us what should be blocked. And actually, there were some things that weren't blocked in other states, because we got recommendations from New Hampshire. But all the natural heritage people are using this portal as well, and they contacted across the board and said, we'd like these species blocked for these states, and they got blocked right away, which is great. It's the administrative level that happens. And then, if you're a bona fide researcher of protected species, then you can contact the curator and be allowed to see those data. But the general public couldn't. And that's something else that I should have pointed out, as my husband said, can anybody do this? That there are people who have administrative capabilities or editorial capabilities, or anybody could search the collections by just going to where I showed on the left-hand menu there, that search collections site. Anybody can pull up those data, except for the things that are blocked because they're protected.